And I want to talk to you about the problem of chronic and latent viral infections. And this is something that, sort of as John alluded to, I've been interested in really since I came into laboratory medicine, both from a diagnostic point of view and then also really understanding the virus. And more recently now, starting to think about what we can do about the virus. So to get us started on this topic, I want to give us an example, sort of the money one to think about, which is HIV. And sort of the unique challenge that HIV presents to us as we think about treatments, and especially as we think about the problem of cure. So the problem of HIV is that HIV infects a cell type, T cells, of which is a subset that have an incredibly long lifespan. It infects central memory T cells, establishes latency in there, and since these cells live for 40 or 50 years, the virus, in theory, could live a very long time. But HIV even does a worse thing. It actually inserts itself into the chromosomes here and puts itself into the chromosomes. We call it integration. So the virus puts itself into this and essentially becomes a part of the infected individual through these central memory T cells. And this combination of integration into a very, very long-lived cell type essentially makes this a lifelong incurable infection. There's no way you can wipe out all those cells. There's no way you can go into those chromosomes and find the viral nucleic acid and do anything very specifically about it. An incurable disease. Now, this is how we've thought about the problem for a long time. In fact, this was the only way you were really allowed to think about this problem for a very long time. But the case I want to make today is that the cure of HIV or other chronic or latent viral infections indeed may be possible, and in fact, I think it should be a research priority. So I'm going to tell you a story, really, and this is the story that really brought this to the forefront and said this is possible. This is the story of Timothy Brown. You've probably seen this guy and his picture in the newspaper. Timothy Brown was infected by HIV. He developed an AIDS-associated malignancy and underwent a stem cell transplant for that. He was cured of his malignancy, but the donor of his transplant had a specific DNA alteration such that their cells were resistant to HIV. So this combination of all the things that we do to people who get transplants, wiping out their own immune system, and then combined with these resistant cells, cured this man of HIV. And so he is the only person that we have a documented proof of cure. 4 years later, he's off antiretrovirals, he has no tumor, and he has no detectable HIV in his blood. So because of this case now, we can at least say it's theoretically possible to cure HIV. So the point is the time is really right to think about curative therapies. And there's really some enabling technologies, and these are what I'm really going to talk about. And these are novel DNA cleaving proteins that I'm going to show you can really find and disable very specific sequences within the human genome. Whether that sequence would be an integrated virus, or whether that sequence would be the genes encoding essential viral receptors, we can recognize them and destroy them. And this raises then the theoretical possibility of elimination of the virus. And doing this, I think, really fulfills what is a logical triad of a response to the HIV epidemic. For a long time, we've been working on prevention, either through behavioral modifications, vaccines, and so forth. We've certainly really made tremendous advances in management through the antiretrovirals that we have now. But really the third leg of that should be the idea of curative therapies. So these things are all going to work together as a part of a response to the pandemic. So here's the outline of the talk. We're going to go over viral latency as a concept. We'll talk about DNA targeting enzymes, really the enabling technology for us. We'll talk about how we want to use these for targeting HIV. And then I want to take a little bit of a side and talk about sort of the big efforts, the big collaborative work that we're doing that John sort of alluded to in the Martin Delaney Collaboratory. You can't do that without talking about targeting CCR5, really the key to curing Timothy Brown. And then at the end, we'll expand this then into some of the other really critical chronic and latent viral infections that are affecting human health throughout the world. So viral latency. We have a few virologists here. You can get virologists going about what's latency, what's persistence, and people will argue. So I want to have a kind of just a simple, loose conception of this. And the idea is that latency is a form by which is a mechanism by which viruses persist for a long time in an infected individual, and they do that by only expressing a limited subset of their viral transcripts and therefore the genes. And because they don't make much stuff, they're essentially resistant or they're invisible to the immune system. So the virus is not replicating, but the genome is there. And you can suppress viral replication The virus could lay dormant for a long time, but periodically the virus can reactivate because the roots of the infection are there and remain. And a lot of viruses do this, and we mentioned at the top HIV. Herpes viruses they're really sine qua non is latency, so we have a list of them there. And then things like hepatitis B and human papillomavirus might be arguably closer to the persistence side of the spectrum, but nevertheless sort of have this same idea of lasting for a very, very long time in a quiescent form. So what would you do about these latent viral infections? How could you clear this? One approach is you could simply try to destroy the latently infected cells. Now, hepatitis B will infect essentially all your hepatocytes. Probably, you're going to need your liver. What would happen if we eliminated all the infected central memory T cells from an HIV-infected person? Certainly could have problems. So that may not always be an option. We could reactivate the latent virus into lytic replication, and a lot of people have tried this for HIV. So the idea is maybe you activate the immune system or you treat with some sort of drug. You're going to make the virus pop out of latency into a lytic replication that'll destroy the infected cells, and then somehow if you could protect new cells from getting infected, the virus would sort of burn itself out in a blaze of glory and be cleared. Obviously hasn't worked yet, otherwise there wouldn't be a talk here, but certainly something people are continuing to work on. And then the final point is sort of the one that we're most interested in, and the idea is to specifically remove or inactivate the latent virus without necessarily having to do any harm to the infected cell. The way we want to accomplish this is through the use of DNA targeting enzymes. And I want to give you a sense of sort of the three main classes of these. Some of them you've heard of. Zinc finger nucleases are probably the best known. There's two other classes, tau effector nucleases and homing endonucleases. Zinc finger nucleases have the structure you see here, and they were originally described as transcription factors. And essentially there are these kind of protein structures, these finger-like structures that are held together by a zinc atom, hence the name zinc finger nucleases. And the zinc fingers, each protein finger very specifically interacts with a triplet of DNA. Okay? And so the specificity of many fingers is known, so you can imagine that if you had a DNA sequence you'd like to recognize here, if you had the right fingers, you could string them all together and recognize very specifically a sequence. Okay, now these are transcription factors. They don't have any cleavage activity, so if you wanted to cleave the DNA, what you do is you hook these things onto a domain of the FOC1 nuclease. This is an obligate dimer to work, so you have to have two of them, and therefore you need a zinc finger on the other strand as well. Okay, so if you make the sequences right to bring those two FOC1 nucleases together, they'll cleave the DNA right there in the middle. Okay. Now you can actually buy these things. You can talk to Sigma, you can pay them money, they'll take a few weeks to months and send you some of these that'll probably work for your sequences. But there's actually some drawbacks. Not all DNA triplets can even be targeted in theory. And there's also what we call context dependence. That is, what one finger binds actually influences what the next one binds, so that not everything you'd think you can target can you really do. And then finally, the biggest drawback of these is that if you can imagine a bunch of FOC1 nuclease floating around here, if this one ever finds this one and come together and form that dimer, they can cleave DNA. So they can cleave DNA in off-target places, or if the recognition is not quite perfect, they can cleave off-target. So they can be toxic, they can cause other problems. The second class are tau effector nucleases. These are really interesting. They were only described a couple of years ago, described in bacteria. Plant pathogenic bacteria, and the idea is the bacteria actually inject these into the infected plant cell. They then find very specific sequences within that and actually alter the transcription of the infected cell. So a very interesting story. And they're based on this 34 amino acid sequence that forms a repeat, much like a zinc finger, except each then repeated sequence binds one nucleotide. Now the code is perfectly known, so you can bind anything you want, any sequence, and there's very little or no context dependence. Okay? So you can make these things to recognize any sequence you want, which is really wonderful. And then essentially you put them together with the FOC1 nuclease and you make the dimers in the same way. These are wonderful reagents except for one problem. If you can imagine that each 34 amino acid repeat recognizes one nucleotide to get a specific sequence, you end up with a very, very large open reading frame of which you need 2 to make a nuclease, okay, 1 for each strand. So they're very hard to actually deliver in any kind of practical way. So mostly used in the laboratory as something easy to make, but not necessarily deliver. What we're most excited about are a class of proteins called homing endonucleases. These are encoded by yeast. They're the protein, functional protein core of selfish genetic elements. And what they do is recognize long DNA sequences, typically about 22 base pairs, very specifically. And if they find them, they cleave them. Okay, so you might think about these things as restriction enzymes, only with very, very large, very long recognition sequences. Yeast use them to induce double-strand breaks, or in yeast they induce double-strand breaks, and the selfish genetic element actually inserts itself into that break through homologous recombination repair. We'll come back to that. But these have some really great characteristics for what we want to do. These are easily the smallest of the 3 classes. They're only about 250 amino acids, very easy to put into delivery vector. Perhaps most importantly, the DNA cleavage activity is actually inherent to the molecule. You don't link these things to a FOC1 nuclease floating around. The molecule itself, the protein molecule, is a nuclease. So therefore, if the binding's not quite right, they don't cleave. Okay, so they're highly specific, they're very non-toxic, and they're just lovely reagents for us to use. The trouble is that what they recognize is yeast sequences. And we need to make them recognize what we want. Now, I'm going to spend a lot of time later in the talk telling you about how we do this, but this has been really an area of great advancement over the last 2 years or so. So we're able to actually redesign these for desired sequences. So how were these things originally envisioned to be used? Originally, people realized that this might be something great for gene therapy, and the idea was based on what they do in yeast. So what we'll do is we'll direct one toward a diseased allele, maybe this red allele here, that we need to fix. Maybe this is X-linked SCID or something, and we'll induce a double-strand break there, and at the same time we'll offer the cell a repair template. So we'll basically get homologous recombination repair right where we made the break, right at the allele. We'll fix the allele. It will be under the proper regulatory elements. There'll be no insertional mutagenesis possible, and we'll have a perfect repair. This was a wonderful idea, but my colleagues who wanted to do this were running into some serious problems. And the problem they were running into is that mammalian cells don't like to go through homologous recombination repair. Yeast love that, but our cells use an alternative repair pathway predominantly called non-homologous end joining. So in non-homologous end joining, the cell finds the 2 broken ends of DNA and basically just takes those 2 ends and puts them right together. Okay? So it's a perfect repair, it's high fidelity, it's rapid, very simple. You don't get other genetic elements swapped in. The trouble is, or not a problem, but if that perfect repair happens in the presence of a homing endonuclease, it gets cleaved again. So it gets repaired and cleaved and repaired and cleaved through multiple rounds until something goes wrong, and then you get maybe deletions on the ends. You get repair. The recognition site is destroyed, there's no more cleavage, but you've also changed the sequence there. You've taken out genetic material. And so a certain proportion is imprecise, you get indels around that. And we reasoned that although this is terrible if you're trying to fix X-linked SCID, if you're trying to destroy an integrated retrovirus, this is a wonderful thing. Because we basically have a reagent that can find a very specific sequence, recognize it, induce insertions and deletions in that region. Okay, so if this is directed toward an essential viral gene, the virus isn't going to work anymore. And so we got very excited about the idea of directly targeting latent viral DNA. Now I mentioned that redirecting the specificity of these things is difficult, and we really didn't know, this is all great in theory, but what would really happen if we had one of these things that recognized an integrated virus? So rather than begin the arduous process of redesigning a homing endonuclease, we said, well, let's do the simple experiment. And the simple experiment is to change the virus so that it has the recognition site for one of these things that already exists. We know how to do it. We do that all the time in the lab. Very simple. And we can ask, if we had such a thing, would it actually work against a virus? So we decided to get a little bit clever. And we said, well, if we're going to do this, let's make a reporter virus. So what we did was to take a GFP sequence here in green, the recognition site for an existing homing endonuclease called ANI1, and we'll put that right in between a translational start site and the GFP. So everything's in frames. The idea is the cells are naturally going to be green. But if we have some sort of deletion event within this, we'll change the reading frame, or we'll delete portions of the GFP or maybe the start site, and we'll no longer get a reporter gene. Okay? So we could very simply follow, is this thing even working or not? Okay? So you can take that, we'll put it into an integrating lentivirus construct derived from HIV. We can then make reporter cells, and lo and behold, the reporter cells are green, just like we thought. Okay? Now, this thing is integrated into the genome. On average, in the clones, one integration event per cell. So the expression is very, very stable. So on the bottom line here, you can see just the expression of green fluorescent protein over time. So this is the x-axis. And you can see that at 3 days, 10, 17, 24, 31. You can keep these things for months, and they'll retain their green. Okay? Now we can take our homing endonuclease, introduce it into the cells, and ask, does anything happen? Now this is, very rarely can you say this in science, as I think a lot of you know, this is like the second experiment we did, and I'll show you, it worked beyond our wildest dreams. So first thing was to introduce an enzymatically inactive enzyme, a control. Okay? So this is something with a mutation at the active site, so it's not going to cleave. We'll link that to a red fluorophore so that now we can follow which cells are transduced or which have the homing endonuclease here on the y-axis. And we'll just look at the top two quadrants and ask, does anything happen to the green? Well, this enzyme is enzymatically inactive, can't do anything, no change. But if we introduce the wild-type enzyme, the one that actually works, we were amazed to see that as soon as 10 days after infection, we got the emergence of this second population of cells that had lost the ability to make a reporter gene. Okay? So this thing was actually in the second experiment we ever did, working to really dramatically reduce GFP expression in the cells. And in fact, when you do later experiments, you tune up your system, you can get essentially complete reduction. We get about a 75% reduction of the number of cells that express a green fluorescent protein. If you remember how this works, a lot of the mutations are frame shifts, so that would be about 2/3. Get a little addition because of big ones that take out the translational start site and so on. So really, anything, as much as we could hope to see in this system. So what are we doing? Well, you can then, if the reporter says you're disrupting the gene, you can PCR that up and sequence it. And I know this is going to be too small for you to see, but I think you can get the gist because on the top line is the wild-type sequence in red. In green is the recognition site for the enzyme. Here's some more wild-type sequence. And so all the places where we have no letters are deletions of genetic material out of the virus, okay, out of the reporter sequence. Occasionally you have some insertions here in blue. And if you sort of look at the distribution, the most common lesion is a deletion, less than 30 base pairs, and then sort of a smattering of other things added together. One thing we worried about is whether this might be unique to a given clone of cells. Maybe sometimes viruses integrate into places where the enzyme can't follow it and cleave the virus. In fact, we haven't observed that. We've looked at multiple clones, and you can see here at the top that you get the same sort of events quite early on. And in fact, even in a bulk line that's not cloned, you can see the same thing. So we really have no evidence yet that there are places viruses can go that the enzyme can't follow. But we'll come back to that later on in later work when we are actually able to test that in HIV. This is extremely well tolerated by the cells. There's no evidence of any toxicity. These are just measurements of cell death. In blue are controls. In red is the cell death in each experimental group. You can see even the ones that have the active enzyme show no evidence of cell death, nor do they have any caspase activation. The only thing that they actually show that's measurable is a slight delay in the G2 phase of the cell cycle, and that's because the cells recognize that they contain a double-strand DNA break. They go through repair pathways, they stop. They stop their cell cycle. Once repair is complete, they progress. But they don't die. So a very well tolerated therapy. What these experiments imply to us is that a homing endonuclease could, in theory, inactivate integrated HIV. But remember, these things only recognize yeast, right? So they recognize yeast sequences, not HIV. But at this point, we realize then it's probably worth going on into the next phases and trying to make something that could recognize HIV. So here's how we redesign the protein. We want to take an enzyme called Y2-Ani1 and re-engineer it so that it can target HIV sequences. Why did we start with Ani? This is the enzyme, first of all, that I showed you in the experiments. We chose it because it's a very active enzyme. It works at 37 degrees at a physiologic temperature. It's amenable to engineering. My colleagues and collaborators had already started to engineer it toward other targets. The crystal structure is known, which you'll see is very important in doing this. And there were examples of success that we could start to build on. And here's a bunch of things that my colleagues in the Northwest Genome Engineering Consortium, NGEC, have worked on. And you can see the progress really over a pretty rapid time course. I mean, 5 years ago, a homing nuclease was redesigned to change 2 base pairs within the recognition site, and that was a Nature paper in 2006. And then you can just see as the time course goes on, things get more complex, more changes that are adjacent to each other, more complex redesigns. Now, 2 changes gets you a Nature paper in 2006. What we're trying to do is change anywhere between 8 and 12 nucleotides within the recognition sequence, so a much higher bar to success. So the first thing we did was to go through all of HIV, all 10,000 base pairs, and look at each 22 base pair window and analyze it on a couple of things. And where we started was to say, what's the likelihood of that 22 base pairs that we can actually redesign a protein for it? And so each one's given a score, and it's based on does that contain motifs that we've engineered toward before in other contexts or that my colleagues have? So we know we can re-engineer for a C at position -6. Or does our computer modeling say it should be possible? Or do we think there's just absolutely no chance? And each one of these things kind of go into an algorithm and get a score. Basically, the lower scores are better. So we come up with a bunch of the entire genome listed out in 20 tumors, and these are the best scoring sites within the genome. Lower scores are better. Here they are. And then we have that list. How well are these sites conserved? Okay. Remember that HIV has a wide variety of sequences, so that entire thing is done on a consensus clade B HIV sequence. So how well are the sites that we picked out conserved relative to the entire world of HIV sequences? So in collaboration with my colleague Tomer Hertz, we did an entropy analysis for each 22 or each base pair within the 22 tumors. So if you're not familiar with this, what you're really measuring here is at each site within the entire HIV genome in blue, how well is that site does have a single nucleotide at it. So if there's absolutely no variation at that site, the entropy score is 0. And if it's absolutely random, it's way out here and past 1.5 or so. Okay. So you can look at polymerase as a whole and say it's reasonably well conserved because everything's kind of over here toward this side. And our anti-target, then, you can see that almost all the sites are very, very highly conserved within that, with only a couple that have a little bit of alteration in them. So we're pretty happy. This is a pretty well-conserved site. What that means is if we made something like this, it would be useful in a wide variety of patients. Or if you put this into a patient, the virus would have a hard time mutating away from it. Okay? And you can do this for a number of them. Here's a TAT target that we ultimately rejected because it actually had a fair bit of variation within its sequence. I like to show this one because it often comes up. We were looking at envelope sequences, and if you look at envelope as a whole, look at how strong the tail is, right? Everybody knows the envelope of HIV is hypervariable. So people say, why do you even bother to look at that? Well, you bother to look at that because there are sequences in the envelope that are highly conserved. And we found a couple of those that are great to target against. This means it's useful against a wide variety of patients, all this. It also means that if we can induce mutations there, they're likely to be of significant fitness cost to the virus because the virus has conserved those sequences for a reason. Okay. So actually we have some envelope targets that we're going for. Now we were real happy that a lot of our targets seem to be pretty well conserved. Later on we realized it wasn't just luck. We had searched against consensus sequences, so we had biased the thing for that. But nevertheless, we were happy to it worked out well for us. We stumbled into success in this case. So based on this, we came up with a couple of lists, a couple of groups of targets we wanted to look at. The first group, what I called ease of design. I wasn't really sure that we could do this at all. So let's just forget conservation. Let's just take the ones that really score the best and make something that can cleave some HIV and prove our principle, and we'll we'll at least have something to go forward with. And then the second group was still had good engineerability, but were also much better conserved, to sort of try to balance those things together. So 2 groups. And then what we do is go through a fairly complex computational design effort that we do together with David Baker's lab. And you know, much of this is the grand we. So if David should see this on UW TV, you know, want to make sure I'm giving credit where it's due. But we co-mentor a graduate student who's actually doing this, Esther Mincer. And what Esther does is to take the wild type sequence here and compare that to the sequence that we want to change the virus to. So this one has, what is this, 9 or so alterations within it. And so for each one of these, Esther actually evaluates how the homing endonuclease here in green is going to fit together with the DNA helix. And this fit of the enzyme in the DNA has to be perfect for this cleavage event to occur. So Esther actually begins to essentially look at the new sequence that we want to recognize here in DNA and actually fit different amino acid residues into that binding area and say what sort of things would work here, and develop a library of potential solutions for this. This is all done in silico at this point. And this is based on a really very important work from the Baker lab evaluating the mechanisms of protein-DNA interaction. And there's a wide variety of motifs, and this is an arginine-glycine interaction, and you can imagine how the molecules are actually going to interact here via hydrogen bonds. Okay, so hydrogen bonding is a very canonical way that proteins might interact with DNA, but there could also be hydrophobic or other types of interactions. So by having a repertoire of these things, you can start to just fit different stuff into this and try to make things that'll bind. And ultimately, you come up with a wide variety of solutions here. These are all on the computer, but when you have a big set of these, you build them into a library and you randomize the DNA a little bit to give it a little more alteration, and then you'll screen them. Now, I'm going to jump ahead of the story just a little bit here and say these are examples of some solutions that Esther's come up with. And these are fun because these solutions that she designed in silico later actually fall out in biology when she does the screens. Okay, so this actually works sometimes. Not every time, and a lot of solutions from the computer actually have no activity at all. But some of them really work. And so by having enough potential solutions, you can come up with real-world solutions. These are then screened in bacteria when you come up with these solutions, and this is a survival screen in bacteria. And it's a 2-plasmid system, and the basic idea is that one of the plasmids expresses the homing endonuclease, or the library of potential homing endonucleases. The other one expresses the target site that you want to engineer toward. These are both inducible, so you can tell the cells when to express them or not. So when you induce this one, you get the homing endonuclease. If these homing endonucleases recognize their target, they cleave this plasmid and destroy it. Okay? So the cells that have a working endonuclease no longer have this plasmid. This plasmid has a death gene on it. So the ones that didn't encode a homing endonuclease that worked, you turn the death gene on, those bacteria die, and the only ones that remain are the ones that have a working homing endonuclease. Okay, so this is the fundamental biological screen, all done in bacteria. And in doing that, now Esther has gone through a tremendous amount of work. I mean, this is summarizing a little over a year's worth of work now in one slide. But to say that she actually has solutions for 4 of these sites that actually show cleavage activity. They'll cleave the HIV sequence. So the wild-type enzyme has now been changed into recognizing HIV. What Esther is working on now is taking these, what we call first-generation enzymes, and really trying to ramp up their activity, which is still not quite at wild-type levels, because the more activity the enzyme has, the better antiviral it is. But she's had a lot of success already in redirecting these. I want to change tacks just a little bit now and talk about some work we're doing with Josh Schiffer on mathematical modeling. And I want to do this because I think it starts to force us to think in a rigorous way about how we might apply this technology as we go forward into some real-world situations. Those of you who've worked in modeling, you know that you need to be careful with how far you push modeling and its interpretation. Depending on the parameters you put in and how well you know them, you can get real-world predictions or junk. But what I think this really is most useful for is forcing us to think rigorously and allowing us to rigorously evaluate what I like to euphemistically call suboptimal experimental outcomes, which means that when we actually do these things and they don't work, we'll be able to work our way backwards and understand exactly why. Okay, what were the defining parameters? So the first thing that Josh did, which I think was very useful, was to try to predict some very kind of commonsensical things. And the first one was, what would happen if we had a homing endonuclease and we put it into patients and we put in one homing endonuclease? Well, we know that HIV, even within an individual, has genetic variability. So you might think that about 1% of the viruses have alterations at that site that would mean it wouldn't get recognized by your enzyme. And Josh's models very clearly pick that up, that regardless of dosing frequency or other parameters, you get a nice reduction of the presence of wild-type virus in the individual, but that 1% quickly grows into the predominant virus within a given individual. Okay? And therefore, total viral load here on the right drops initially and then rebounds. This is exactly like what you'd see with monotherapy with HIV. Nothing too surprising. But it did say that the models at least reproduce some common sense work that we've seen in other situations. But this is actually something that we were interested in, and say, well, how many homing endonucleases do we really need? And so Josh was able to provide sort of some initial estimates on that, and they were rather encouraging for us. And so, we made some assumptions here, all of which are up for argument, and we can actually figure out which ones matter. We'll assume a delivery efficiency of 50%. That is, we try to get virus into a cell, it works half the time. The sequence termination is 99%. That is, if the homing endonuclease is in there and has a target, 99% of the time it causes a mutation. That's based on our data. And the baseline resistance is 1%. And what this shows is that with as few as 2 homing endonucleases, you can get eradication of the virus in a model system. Now, for the aficionados, this actually includes homeostatic proliferation of the reservoir, which is probably worst case scenario, actually. So 2 is the magic number. I like 3 personally. I want to be conservative. 3 obviously has precedent, again, in the antiretroviral therapy world. But it says we're not going to need 20 of these things. Okay. But what the modeling then does tell us is that what's really going to be critical is the delivery and the dosing. That is, what really matters here is how many cells can you really get these things to? How efficiently can you get them to the cells where it needs to be? And so if we look at sort of various parameters here, again with similar assumptions, but now saying what happens if we can only target 10% of the cells, 30, 50, 90. If we can only hit 10% of the cells per delivery, it really doesn't matter at once per week dosing how long we do this. We'll never really get eradication. But interestingly enough, Josh suggests that if we dose more frequently, we probably can overcome the homeostatic proliferation of the virus. So, the harder we push these, probably the less real world they are at this point. But this is going to allow us to interpret the data we get as we start to move into preclinical models. Now, one thing that fell out of the models that was useful to us is that the efficiency of termination of the sequence is critically important. That is, if the homing endonuclease gets in, it's very important that it causes a mutation. And so we knew that was working reasonably well, but some of these cells are good at repairing their DNA, particularly healthy primary human cells, and I'll show you data for this. So they can kind of keep repairing until the homing endonuclease goes away, just expression ends, and therefore nothing happened. And we recognized that this was a problem, and then this paper came to our attention. And this paper is about a molecule called TREX2, and TREX2 is an exonuclease. It's a 3' exonuclease, and the homing endonucleases, when they make their cleavage, they leave sticky ends, like a restriction enzyme, and homing endonucleases, they're 3' overhangs. For the others, they're 5', but for this, it's a 3' overhang. And so if you include a 3' exonuclease, we reasoned, as soon as the double-strand break happened, the 2 free sticky ends would be blunted, essentially, just like you would do in molecular cloning, and therefore you couldn't get a precise repair anymore. You'd always get mutation. So we said, what would happen if we put these things together? And in fact, when you co-express these things, you increase your frequency of mutagenesis, just like we predicted. And this is done in Hep2 cells. This is pretty much the same experiment I showed you before. So here's our green. This is the inactive enzyme with or without TREX, so no difference, no second population. Now this is only 8 days. This is very early. So with the Y2, you start to see a little bit, but the Y2 plus TREX actually very efficiently induces mutations. Everything seems to be working a lot better. And you can see, if you look down here, the frequency of mutation in the Y2 plus TREX certainly is higher by sequencing than it would be otherwise. Now where this mattered went back to a question that I got a lot in these talks. And I'd show sort of the data that I've shown you up to this point, and people would say, well, how do you know, you just showed me Hep2s, that's a tumor. Well, is this going to work in a primary cell? And I go, oh, sure, we think it'll work. And the answer is actually it doesn't work as well because healthy cells are really good at repairing their DNA, right? I mean, we repair double-stranded DNA breaks very well. Tumor cell lines have aberrant DNA repair, so they're much more mutagenic. So when we do the same experiment in essentially a primary cell, we still wanted to be able to work with them, so we turd immortalized primary fibroblasts. So we could work with them, but the DNA repair is going to be healthy. And now if you do this experiment of same control, it's going to show the same thing. If you do Y2 alone, you really have no mutagenesis at all after 12 days. Okay? So really nothing. These cells can repair their DNA as long as they need to. But if you do this together with TREX, you can see now a much higher induction of mutagenesis because as soon as those breaks happen, they get trimmed off. Okay? And that's confirmed by sequencing, where here if we look at the Y2 alone, we really have almost no mutation at all, and at least a third of the sequences that we looked at were mutant after only 12 days. Okay? So this is really critical, and now going forward in our experiments, we actually are co-expressing these almost always together. All right. I know some of you are interested in kind of how are we taking this forward into starting to make this a real thing. And so I wanted to just sort of mention the Martin Delaney Collaboratory that we were awarded earlier in the spring. NIH put together, NIH decided that it was time to work toward cure, and they decided that this was going to be a complex undertaking. This would take the collaboration of large groups of people. So they opened a competition for this that was very well funded. They wanted to fund one or two collaboratories. And we put together a project, myself together with Hans-Peter Kiem as my co-PI. And we really wanted to build upon the strengths of Seattle as we thought about curing HIV. So we thought about hematopoietic stem cell transplantation that's been pioneered at the Hutch and really put the Hutch on the map. Genome engineering, the sort of stuff that I'm talking about here, the Northwest Genome Engineering Consortium, and then all the sequencing and HIV sequence analysis that Lisa's done and Jim Mullins, and really areas of tremendous strength for Seattle, and build something that was really coherent that could work on these. So structure diagrams probably don't really matter for this audience, but I do want to show this one that if you're ever contemplating putting one of these things together. NIH said, oh, we want these things to be really collaborative and truly work together. And so we put together a project where, that has 5 projects and 5 cores, and each one of these lines represents the fact that this project in circles interacts with, some other project at the core. And this is not bogus. So this is like, these projects and cores truly work together tremendously. And when we were reviewed, that was, very positively commented on. This is a very interactive program. Now, I want to explain to you what we're doing, but I can't do that without going to the other thing that one might do about HIV, and that is you might target CCR5. And CCR5 is a co-receptor for HIV. So many of you may know that sort of what we consider the receptor for HIV is the CD4 molecule. This is why it likes to go to T cells, why it can also go to some macrophages. It needs CD4 to enter. But that's not enough. And the virus actually needs a co-receptor. It either needs CCR5 or CXCR4, kind of shown here, and the combination then allows the virus to fuse and enter. Okay? So they're absolutely required for HIV entry. It turns out that CCR5 is essentially irrelevant to human health. Okay? So there's a subset of people who have alterations in CCR5 such that they don't make any functional protein. Okay? They're completely healthy. Maybe a little bit more trouble with West Nile virus, a couple of other things, but pretty minor stuff. So CCR5 is largely dispensable for normal function and development. CXCR4 is not dispensable. If you don't have CXCR4, it's embryonic lethal. It's required for development of multiple cell types, but not T cells. So actually, one could think about knocking this out of T cells, and that may be something to go forward. But the principles of CCR5 knockout have been demonstrated by experiments of nature, because there are people who don't have it, and this is how Timothy Brown was cured, because the cells that he got were null for CCR5 expression. Okay. So one thing I want to point out, we're not only thinking about HIV here. This is unique to HIV. This idea of there's a co-receptor that we don't need is amazingly good fortune in the fight against this virus. This does not exist for other viruses. It's truly an Achilles heel of this virus that we're going to take advantage of. And so, this is the New England Journal report about Timothy Brown. So this would be great, but you can't do this for everybody. In fact, the first paper was in New England Journal 4 years ago. Everybody would love to be second. And nobody can pull this off again. And the reason is it's already hard to do a stem cell transplant. You have to match for HLA. It's difficult to find donors. You have to look all over the world. Less than 1% of people in advantageous ethnic groups for CCR5 are homozygous null. Okay? So you've got the difficulty of finding a CCR5 donor times 0.005 means nobody can actually find a way to do this again. Okay? And we estimate that less than 0.5% of patients would this even be possible. Okay? So what we would like to do in the collaboratory then, in this part of it, is to think about autologous transplantation, where we could use these same genetic targeting approaches that we've been talking about, targeting toward the virus, and target it towards CCR5 in a T cell or a stem cell, knock that out, and use that to replenish the virus. So do Timothy Brown's experiment, but in a autologous setting. So here are the projects. The first project is Ann Wolfrey's work, and she's actually asking how much did the conditioning matter. Timothy Brown actually had 2 transplants, so he got very, very heavy chemotherapy. Wiped out a lot of the reservoir by itself, we're sure. So we don't know how, it was an N of 1, so we don't really know why he was cured. So we're doing that. We're working with Sangamo Biosciences, that's Philip Gregory Project 2. They are the zinc finger company. So, I said some bad things about zinc fingers, but, in fact, you can make them. They're being improved all the time, and they're being used to knock out CCR5 now. Hans-Peter Kiem then is taking those and asking whether that can be used to control HIV. And so actually taking those zinc fingers. At the same time, in the other half of the project, we're trying to knock out the integrated virus itself. Okay, so we're actually going to knock that out of stem cell products or in vivo, actually in infected patients. And then the final project is working with John Rossi from City of Hope in California to help us come up with more efficient ways of actually targeting these things where they need to go. And John's an expert on delivery of siRNAs using aptamers, RNA aptamers, and PAMAM dendrimers. And had a very nice paper showing that if you deliver siRNA using these aptamers, you can knock down HIV in an animal model. And so, we went to John and said, well, if you can do that with siRNAs that only exist for a short time, have we got a payload for you that's going to knock out the virus permanently? And so John's very excited. So these are all working together. Hopefully you'll get a chance to come back maybe and give you an update in a couple of years and see how much progress we've made. In the last just couple of minutes, I want to turn. We've talked about HIV. HIV is unique among human pathogens. There aren't that many that actually insert themselves routinely into the chromosome, like we talked about, that integrate. Most viruses that were sort of on the list we mentioned earlier exist as episomes, circles of DNA that are maintained within the nucleus that replicate with the cell or stably maintained. So we asked, could we do anything about those sorts of infections as well? So, we'll just take our same reporter construct, put it into an episome and establish cell lines, and do the same sort of thing. So now we get a nice stable population of cells, but they now essentially contain an episomal target rather than an integrated target. you put in the inactive enzyme, nothing happens, just like I showed you before. But if you put in Y2 anti, you get a very dramatic and very complete eradication. And we think that what happens here is that if the episome is cut, it's often just lost. Not only is it mutated, but it just degrades. And so you truly do get a cure of these cells from the target. This opens up the possibility to think about some other infections, okay? So we've talked a lot about HIV, but hepatitis B, an enormous problem in the world, herpes simplex, human papillomavirus, all persistent, long-lived infections that cause significant health problems. And when, if we think about everything from sort of, a little egocentric view of the world here. So if we think about these things from our point of view working on this, does each virus have characteristics that are good for us or bad for us? And HIV has some drawbacks. I mean, clearly, if you want to work on it, the disease is bad, so it's important. And there's a lot of funding interest, which has been great, okay? HIV has some difficulties, though, as well. HIV is extremely genetically variable, right? So we got to deal with that. But there's really two very difficult problems with HIV. One is that delivery is going to be hard. The latent reservoir is 1 to 10 million latently infected central memory cells scattered throughout the body. They're quiescent. How are we going to get the things there? We'll talk about that at the very end. And then the second problem is, if you really want to cure somebody, you probably have to get rid of darn near every last one of those latently infected cells. Because if you don't, the virus can use that to reestablish itself, okay? So the need for complete eradication, very important. Now, some of these other viruses are actually much more advantageous in that view. I mean, in fact, for every one of these other infections, there's a lot more people who are infected. Many of them are very serious. There's 400 million people chronically infected with HBV in the world, okay? We think this is a solved problem because there's a vaccine. 400 million people, okay? Many of whom are going to progress to cirrhosis or hepatocellular carcinoma. It's a huge health problem. A lot of genetic variability there, but delivering things to the liver, to the infected hepatocyte, is easy, okay? We have many, many ways that have been shown to work for this, okay? So we're very excited about that. HSV, very little genetic variability we have to deal with. Maybe the disease isn't so bad, maybe there's not as much funding interest, but, delivery is easy, and, for all these, we may not actually have to completely eradicate the virus to make a huge difference. We know that if you knock HBV down below a certain level, the immune system can often control it. Josh has done some really lovely modeling in HSV in collaboration with Larry Corey and now with us, which suggests that if we could reduce the viral load within the dorsal root ganglia, which is, there's a dorsal root ganglia contains 10,000 neurons, okay? And this is where all disease comes from. And of those 10,000, only 100 are infected with HSV. And all disease comes from those 100, okay? So if we can knock that down to 10, we can make an enormous difference in clinical manifestations of disease, okay? So, these have some really huge advantages. The same thing about human papillomavirus, clearly a very, very serious disease, and it really should be 3 pluses because you think about, cervical cancer and head and neck cancer. So huge, huge problems, all of which I think are very advantageous or have some certain advantages. So we have active programs in all these. I just want to show you really quick. Hepatitis B, we're designing homing endonucleases for this. The completely different approach, actually. We're hedging our bets. This has little or nothing to do with rational understanding of the structure. This is a high throughput screening-based approach where we've actually just working together with Barry Stoddard and Andy Scharrenberg have worked to just basically divide the entire recognition site up into what we call modules, randomize those and ask if we had a totally randomized library of homing endonucleases, which ones can be cleaved or not, and then develop those libraries and then basically just look through different sites of HBV and ask, are there solutions that exist in nature? And for some of these, like this S10 site, there were solutions for everything we need. We can then go back into these huge libraries and pull them out and say, what are the solutions, and start to build them together. So we don't need to know the structure. We just know they work individually, and then we can put them together. It actually seems to be working very well, and we have one called S10 that's targeted here in the HBV genome. We also have 3 additional zinc fingers that we're working on as well. So we'll actually be able to compare all these things that I told you. Maybe this toxicity problem isn't so big, and maybe the zinc fingers will be the answer. So we actually have a handful of reagents now that will be able to go into some really elegant mouse models in collaboration with Lauren Terrell in Alberta to test these things in vivo within the next few months. The last idea, delivery of these things to the target cells. How do you get them there? This is the money question. Clearly the most, I mean, when we started this, right, we didn't know whether these things would be of any use against a virus, okay? Okay, that was solved. These would be great to have. Then we didn't know, well, can we ever really redirect them? And that's gone faster than I ever dreamed, and the answer is, yeah, we can redirect them, Now we got to get them where they need to go. And this is the one that's always scared me. So I'll tell you that, there's a number of ways they work in culture, and under the right situation, clinical situation, they're applicable. So you could liposuct cells if you've got stem cells in a preparation that you want to transplant. Liposuction, electroporation, those all may be good things to do. We're working very hard on viral vectors. All the experiments that I showed you actually used lentiviral vectors. We've used integrating vectors and more applicable non-integrating vectors, ones that will be very safe. They work very well. We're very, very excited about adenovirus and AAV. And I just hired a really talented scientist into the lab, Daniel Stone, who's going to help us. Wonderful vectors for getting things to the liver. Okay, great for hepatitis B. Also very, very good at transducing dorsal root ganglia. You can actually inject these things into dorsal root ganglia and get nearly complete expression of your transgene. So very exciting. Herpes simplex is a fun vector because it loves to go down axons to the innervating dorsal root ganglia. So we're going to use engineered herpes against herpes. And at length we could talk about the tricks we go through to make sure this is non-pathogenic, but very potentially elegant way of delivery. We're working eagerly. I mentioned aptamers. There are RNA aptamers. There are also DNA aptamers, and we're working with Jeff Baird. I don't know if Jeff's here, but to work on those. And so those are all really great for a variety of ways. Will those work for HIV? I don't know. We're going to evaluate this. HIV specifically, we're actually interested in what is, I think a little bit forward-looking, maybe out there prospect, but we want to think about whether we could use dendritic cells as a Trojan horse, as a delivery vehicle. If you remember, what does central memory T cells do? They sit around and they continually interact with dendritic cells and say, do you have my antigen? And no, no, no. And if they ever find the one that has an antigen, then they cause a memory response. What we'd like to do is engineer dendritic cells to basically go around and hand these things off to the central memory cells as this normal interrogation happens. So we'll come back and tell you whether any of those things worked maybe in a couple of years. So to conclude, I just want to make the point to you that the paradigm shift here is that the latent viral genome itself can be a valid therapeutic target. That we don't just have to think about blocking replication and sort of holding things down. You can actually think about going after the latent genome itself, and that's sort of the money. That's the root of the infection. That's the root you got to pull out of the garden. Okay. For HIV, for all these, but especially for HIV, the big challenge is going to be delivery. If we can crack that, then I think we're home free. But this is going to be the hard one over the next many years. And then finally, for my colleagues who work in other areas, I really do see this as a complementary effort to all the other things, and we're going to need all these things really to address the HIV pandemic and these other infections. I want to finish up by thanking the people who've been involved. In the left top are the people in my laboratory, just in alphabetical order. I think I've mentioned a lot of their work and them by name maybe as we went along. On the right, a number of collaborators. This is truly collaborative science. You've heard we a lot, and we is involved. All these people, I hope I've mentioned most of their names in the appropriate places. But these sorts of experiments are just bigger than any one person. And to understand really all the technology that's needed to move it forward, it truly requires a collaborative effort. And then at the bottom are the funders, many of whom were with us when this was so speculative that people, the NIH certainly wouldn't look at us at the time, and now we've progressed, I think, to where it's getting to be a little more mainstream, and we're grateful for all the support from everybody.